Our lab was the antimicrobial efficiency testing of Crest, Pro Health, and Listerine with and without alcohol against Staphylococcus aureus and Klebsiella pneumoniae, and investigating microbial resistance due to prolonged exposure. And this is by Daniel and Heather. So our oral cavity is home to many microorganisms, and it's one of the most diverse microbial communities in the human body. While some of these microorganisms are relatively harmless, many can cause oral infections and diseases. Many mouthwashes today contain various antimicrobial agents which have shown great success in killing oral microorganisms and preventing the development of many oral diseases. However, it is important to note that prolonged use of mouthwash has the potential to lead to the development of microbial resistance. Microorganisms can adapt to the antimicrobial agents present in mouthwash and become less susceptible to their effects. While mouthwash has become an effective tool in maintaining oral health, we use it judiciously to prevent the development of microbial resistance. Our reasoning for studying this lab was due to the fact that antimicrobial resistance is a growing concern that we need to investigate more, particularly in the context of mouthwash use. When microorganisms are exposed to antimicrobial agents such as these found in mouthwashes, sublethal concentrations may not kill them outright. Instead, their growth and replication is slowed down, which provides an opportunity for the microorganisms to adapt and evolve resistant mechanisms. This can ultimately lead to the development of antimicrobial resistance and the emergence of superbugs that are difficult or impossible to treat with existing antimicrobial agents. So our first hypothesis was that the antimicrobial efficiency of mouthwash against oral pathogens will decrease over continuous exposure due to the development of resistance. This hypothesis is based on the principle of antimicrobial resistance, where microorganisms can adapt and develop resistance to antimicrobial agents over time. Our second hypothesis is that the mouthwash with alcohol will show a greater efficiency in reducing the growth of oral pathogens compared to those with no alcohol. This hypothesis is based on the fact that the alcohol has been shown to have antimicrobial properties and can effectively kill a broad range of microorganisms. Our final hypothesis was that mouthwash with active ingredients cetipridinium chloride will show a higher efficiency at inhibiting the growth of oral pathogens in comparison to the mouthwashes with sodium benzoate. This hypothesis is based on the fact that cetipridinium chloride is a well-known antimicrobial agent that has been shown to be effective against a wide range of microorganisms. The materials used in this lab were TSA purity plates, which are a general-purpose medium growth used to cultivate a wide range of microorganisms, including bacteria and fungi. They are used to isolate and identify individual bacterial colonies. The bacteria we used in this investigation were S. aureus and K. pneumoniae. These are strains of bacteria that are commonly used as model organisms in microbial research. The plates we used for the test of efficiency of mouthwash were MHA, also known as Muller-Hinton agar. This is a standard growth medium used for antimicrobial susceptibility testing. It has a low nutrition content and a uniform surface, which makes it suitable for testing the susceptibility of organisms to antibiotics in mouthwash. The broth we used in between each generation to rehabilitate the bacteria were lecithin broth. It contains soybean meal and lecithin, which serves as nutrients for bacterial growth. Antimicrobial mouthwashes were used, including Crest Pro Health with and without alcohol, and Listerine with and without alcohol. These are oral hygiene products that contain antimicrobial agents or essential oils. They are used to reduce the number of bacteria in the mouth and prevent oral infections. Lastly, we had a control MHA plate. This was a plate that is inoculated with bacteria but does not contain any antimicrobial agent. It is used to verify the purity of the bacteria culture and to ensure that the antimicrobial activity observed in the test plates is due to the mouthwash and not to any contaminants. The first step of this experiment was to streak the TSA purity plate with the two bacteria strains used in this investigation. They were incubated at 35 degrees for 24 hours. Next, the MHA and lecithin broth medium was made and sent to sterilization by autoclaving to ensure that there was no contaminants that could affect the result of the experiment. The next step was to test the 4 antimicrobial mouthwashes, which were 2 variations of Crest Pro Health and 2 variations of Listerine. The purpose of this experiment was to determine the antimicrobial efficiency of those mouthwashes against the 2 bacterial strains. To do this, each plate was carefully labeled with the name of the organism being used and the type of streak being employed, and which quadrant was which mouthwash. After this, each plate was streaked with the respective bacteria and left to dry. One set of bacteria was streaked for long growth, while the other was used as spread plates. After dried, 4 wells was carefully made by punching the holes into the agar with the end of a sterilized pasture pipette, one for each plate. After each well was made, 30 microliters of the respective mouthwash was pipetted into each well using a micropipette. When this was done, the plates were carefully taped together, right side up, and incubated for 24 hours at 35 degrees. After 24 hours, a loopful of each bacteria was inoculated into lethan broth and again incubated at 35 degrees for 24 hours. Once the bacteria had grown in the broth, 0.2 milliliters of each broth was added to a 9.8 milliliter of phosphate buffer, allowing for a 2/100 dilution. Once done, new MHA was again streaked, and the process was repeated over 4 generations.
After incubating the MHA plates, we then measured the zones of inhibition and selected specific plates that contained the best data within the parameters of our experiment. Table 1 shows the generational growth of Staphylococcus aureus and Klebsiella pneumoniae, lawn growths that were exposed to Crest Pro Health that was free of alcohol. It can be seen that there are acceptable results for both species of bacteria between G1 and G2. S. aureus further managed to reduce its zone of inhibition within generation 3 compared to generation 2. However, as can be seen, the zone of inhibition diameter seemed to plateau for K. pneumoniae at generation 2 and generation 3 for S. aureus. Therefore, based on the results of the data shown in the table, it can be said that Crest Pro Health consistently reduced growth across 3 generations for S. aureus and 2 generations for K. pneumoniae, while Listerine showed no effect. We also looked into the active ingredients of Crest Pro Health, which are, as Heather said, CPC and sodium fluoride, and found that they are effective in reducing oral pathogen growth. And to visually represent our results, we've included 4 figures. Figures 5 and 6 show the generational growth of K. pneumoniae, while figures 7 and 8 show the generational growth of S. aureus. It is shown in the figures that the overall reductions of the zones of inhibition between the 2 generations of both species of bacteria was substantial. As with any study, there were some possible sources of error that we considered. The first possible source of error can be attributed to the possibility of the mishandling of the plates. During our experiment, we had labeled the plates to emphasize that they should be kept in an inverted position. However, we should have also stated that the plate not be moved sporadically during transfer to and from the incubator and fridge. This shift and spilling of the mouthwash is most likely attributed to the distortion of the plates and overall data. And a second possible source of error could result from incompatibility of mouthwash antibacterial ingredients with the selected organisms, as exemplified by the bacteria being unable to grow properly when exposed to Listerine. Thirdly, it is possible that too high of a volume of mouthwash was utilized. 30 microliters of mouthwash resulted in filling of the well almost above the brim. Therefore, it would be recommended to use around 25 microliters to maintain the reproducibility of the results. While we were disappointed to encounter some inconsistent results during the study, which can be clearly exemplified by the scientist who is so obviously frustrated at his inconsistent results, we were able to establish that the study proved that Crest Pro Health overall is more effective in reducing the growth of oral pathogens, while Listerine Care is not. Finally, we do recommend further research to be conducted on the effectiveness of other mouthwash brands and their active ingredients in reducing the growth of oral pathogens, not only S. aureus and K. pneumoniae, but other possible bacteria that can inhabit the oral microbiome.